Hello. Today our chapter is Histology of Tongue. So, tongue is a highly muscular organ located in the mouth cavity and distinguished into free oral part and root. Dorsal of the tongue is rough while its ventral surface is smooth. The bulk of tongue is made up of striated muscle fibers, of which are grouped into bundles disposed in longitudinal, transverse, and vertical planes. The muscles are embedded in loose fibroelastic and adipose tissues covered with a mucosal membrane or mucosa. Mucosa is composed of striated ischymous non-keratinizing epithelium. Internal to it is a connective tissue layer, the lamina propria, which is closely bound to the underlying striated muscles and is very thin in the ventral aspect of the tongue. The mucosa covering this side exhibit numerous little projections called lingual papillae. These are small organs and distinguished from the connective tissue papilla, which indent the stratified ischymous epithelium. Each lingual papilla consists of a connective tissue core. The primary papilla made up of lamina propria covered by the stratified ischymous epithelium. The primary papilla gives rise a small sensory papillae which push into the epithelium. According to the shape, the lingual papillae are distinguished into four types: filiform, fungiform, circumviolate, and foliate, among which filiform is one of them. So, filiform papillae, the name derived from the phylum, which is nothing but the thread. It is the most numerous and evenly distributed in rows over the entire dorsum. Each is a tall, narrow, conical structure composed of both lamina propria and epithelium. The epithelium alone also forms one or more secondary projections which taper into thread-like points. The whole papilla is bent towards the posterior side of the tongue. These papilla are specialized for tactile sense and have abrasive action. Taste buds are absent in these papillae. The next of papillae is fungiform papillae. The name itself indicates that fungus means mushroom-like. These are scattered unevenly among the smaller but more numerous filiform papillae. They are so named because they appear like small mushrooms which are narrow at their base and have a short stalk and expanded, smooth, flattened, rounded top. Each has a central core of lamina propria from which secondary papillae project into the thin covering epithelium. The papillae are richly supplied with blood vessels and may contain few small taste buds. The next type of papilla are nothing but the circumviolet or violet papillae. Their number varies from 7 to 9 in human tongue. They are restricted to the posterior region of the free oral part. Each is surrounded by a deep circular groove. Into the bottom of the groove open the duct of certain lingual salivary glands. These papillae are the largest of all. They have broad flat free base and narrow free surface and narrow base. Each circumviolet papillae has a central primary papilla and several secondary papilla projecting upwards but usually not into the side. Surface of each papilla bears numerous taste buds. Now the last papillae is nothing but the foliate papillae. The name itself indicates that it is looks like folds of the pages of the books. The mucosa is characterized by prominent secondary papillae. Each foliate papillae occur in groups along the dorsolateral margin in the posterior portion of the free oral part. They possess well developed taste buds on their sides. The taste buds are the organ of taste or gustatory organs located along the sides but never at the exposed surface of the lingual papillae. Under light microscope, they appear as small, pale, barrel-shaped bodies embedded in the epithelium. They extend from the basal lamina to the surface. The epithelium over each taste bud has a small taste pores. Three types of cells occur in the taste buds. The types are the most numerous peripheral and curved supporting or sustentacular cells. They have a uniformly elongated shape and pale round or oval nucleus. Second type of cell is type second cell of neuroepithelial or sensory or gustatory cells occur in the central position. Each cell is long and spindle shaped with a bulged mid portion containing dark nucleus. The type second cell has extensive small endoplasmic reticulum. The third type of cell, type third, is similar to that of type first cell, also identified in the taste buds of rabbit. Basal and peripheral cells also described, which is progenitor cells of the taste buds. Under light microscope, only gustatory cells show sensory hairlets or taste hairs projecting into the taste pores. These are now recognized as clusters of large microvilli under the electron microscope. Moreover, they occur at the tips of both type first and second cells instead of sensory function. Typically, synapses of nerve fibers with the taste bud cells have been observed in type third cells. Because of these recent findings, the supporting and sensory cells, also called gustatory cells, the lingual glands also of three types, which is tubulo-alveolar salivary glands embedded in the tongue musculature. They are anteriorly mixed type, posterior they are mucus in nature, and gustatory they are serous in nature. All ducts and the gustatory glands are the ones whose ducts opens into the groove and around the base of the circumvallate papillae. So in such a way, the tongue is also one of the most important organ in the digestion, which is helpful for the movement of food material and formation of the bolus of the food, also helpful for the mixing of the saliva to the food particles.